Hello everyone. Topic of discussion for today is central dogma. As we all know, central dogma is the process in which the imprinted informations in the DNA are converted into functional product. It was discovered in the year 1958 by Francis Crick, who discovered DNA. It is an important element in molecular biology which explains the flow of information from DNA to RNA to make a functional product, that is protein. So basically, in central dogma, we will be reading three important processes. Replication, in which DNA is converted to its own copy. Transcription, where DNA is converted into RNA, and translation, where RNA is converted into proteins. That means the informations encoded in the RNA are converted into proteins. Let us begin with the very first process of central dogma, replication. Process of replication, as we all know, it is a copying process of DNA. It requires multiple sets of enzymes which are acting with high accuracy. So if any error occurs in the replication process, it will lead to mutation. We'll be discussing about mutation in the next part. Let us discuss some of the important functions of the enzymes involved in replication. Helicase. It is the very first enzyme. Helicase helps in the unwinding of DNA duplex. When DNA is opened, during that time, unwinding happens with the help of helicase enzyme. Topoisomerase. So what happens? We know that DNA is a double helical structure. So if DNA is double helical structure, so usually what happens when unwinding takes place, there exists a kind of tension in both the strands, due to which both the strands may get split into pieces. So in order to avoid that, topoisomerase are involved. They relieve the tension when two strands are getting uncoiled. SSB protein, which are single cell binding protein. It stabilizes the single stranded region. What happens when helicase unwinds the DNA? It moves ahead. So in order to avoid the recoiling process, SSB proteins are very important. They attach to the uncoiled areas and avoid the recoiling process. Next, we have primase enzyme. These are DNA-dependent RNA polymerase enzyme. Primase enzyme help in the synthesis of RNA primer. Primers are small nucleotides of RNA. RNA is a single-stranded structure, and there are some RNAs which are made up of very small stretch of nucleotides. Some of those nucleotides can be categorized under primer. Next we have DNA polymerase 1. DNA polymerase 1 is an enzyme which is DNA dependent. So it helps in the erase of the primer. When entire replication process gets over, the primers are removed by the same DNA polymerase enzyme. You can relate primer, the attachment of primer with a knot. While making a garland, we generally put a knot at the end of a string, just to avoid the beads or the flowers to get off from the other side of the string. So in order to avoid that, we put a knot. So we can relate the primer formation with that knot structure. So when replication is about to start, primer is very important because it helps in the process of continuous formation of the nucleotides. But when the replication process gets over, the primer has to be removed, and the same is fulfilled by DNA polymerase 1 enzyme. Next we have DNA polymerase 3. DNA polymerase 3 is the most predominant enzyme because it is the same enzyme which synthesizes the entire DNA. So the replication process is aided by DNA polymerase 3 enzyme. Since it is a complex structure, so just going to share some of its very basic structures. DNA polymerase 3 is actually a dimer structure, and the functionality of this enzyme is that it replicates both the strands simultaneously. It consists of 3 basic units: catalytic unit, proofreading, sliding cam. You can also call it as alpha, eta and beta unit. So these all will be discussed when the structure of enzymes will be dealt. Next we have DNA ligase enzyme, which we commonly called as ligase. It joins the end of the DNA. So when this also will be explained during the mechanism. Okazaki fragments are formed. These are the small stretches of DNA which are left behind in one of the strands of the DNA after the replication process. So this has to be joined together to get the continuous stretch, and that continuous stretch will be achieved by ligase enzyme. It joins the small fragments which are called Okazaki fragments. Then we have few more enzymes. Nucleus enzyme. It is called a repair enzyme because it helps in cutting off the defective nucleotides. If suppose during the replication process any wrong nucleotides are involved, so it may lead to mutation as explained earlier. So in order to avoid that, it checks the entire stretch of the DNA after the replication process. If any nucleotides are mismatched, it will remove those. Now the last we have phosphorylase enzyme. From the name itself you can just understand it helps in the phosphorylation. Phosphorylation means addition of phosphorus, right? So here it helps in the phosphorylation of nucleosides. Sorry, it should be nucleosides. Nucleosides means having nitrogenous bases with sugar, basically pentose sugar. So when nitrogenous bases and pentose sugar combines, they form nucleosides. But in order to energize these elements or these components, we have to add phosphorus, and the addition of phosphorus to it will convert nucleosides into nucleotides, which are the final components of the DNA structure. So the addition of phosphorus to nucleosides is aided by the same phosphorylase enzyme. So I hope you have understood till this. The next part of the lecture will be the replication machinery or the process of replication. Thank you.